About the plasma membrane in the last lesson, and can anybody tell me what are the main components of the plasma membrane? Phospholipids, protein, cholesterol. So these and carbohydrates. Remember, some proteins are glycolipids. So the plasma membrane is made up of several components, and most of the function of the plasma membrane comes from what? From the proteins. Okay, so remember we listed several different type of proteins, like enzymes, receptors, transport molecules, recognition molecules, and so on. Okay, so the plasma membrane, most of the function comes from the proteins. Now in the next few lessons we are going to talk about the transport mechanisms, how the cell uses the plasma membrane to transport molecules in and out. And there's many different mechanisms, we'll discuss them. Some require energy, some don't require energy, okay?
Yeah.
So before we start on that, we need to define a few things. Number one, what a solution is, okay? So, you probably learned this in chemistry. If you get some salt and put it in a glass of water, okay?
Yes. Yeah.
The salt dissolves, okay, in the water. It makes a solution. That solution contains the solute, which is the dissolved substance, okay, the salt in that case, and the solvent, the dissolved meaning, which is the water, okay.
Yeah.
So that's what the solution is, basically dissolved solutes in a solvent. So like, remember in the lab we did that experiment with the tonicity. We had the different concentrations of the salt, and you put the red blood cells. So basically those are different solutions, okay? So in terms of concentration, we're talking about the amount of solute in that particular solvent. So the amount of ions of that solute, okay? So the amount of salt in that solution is the concentration of that solute, okay, or that molecule. So it's obviously, in this case, it is in water. So if we have a higher concentration of solute, that means the water concentration will be low. And remember we talked about tonicity in the lab?
Yeah.
So we're going to talk about it again by the end of this chapter. But remember, tonicity is the concentration of the solute. So if we have a hypertonic solution,
Solute is more than salt.
more solute, correct?
Yeah.
Less water.
Then it shrinks.
So what about hypotonic?
More solid.
Less solute.
Okay.
More salt.
More salt.
And more salt.
Less solute, more
salt.
hypotonic, less solute, more water.
Salt. Yeah.
Okay? And in our body, what we have is isotonic conditions where there's equilibrium between the inside and outside of the cell. Okay?
Okay.
Not always, okay? But in because we need these concentration gradients as well in our body to transport molecules, okay? So what we call concentration gradients, okay? Before I move on, obviously, as I mentioned, hypotonic. So pure water containing no solute would be hypotonic. Means it has no solutes in it. It's 100% water. So it's hypotonic. Okay?
So pure water is hypotonic?
Yes. You got that?
Got it.
In terms of concentration gradient, now this is you establish concentration gradients between two regions that are next to each other and that are separated by a barrier or a semi-permeable barrier. In this case, we have the inside of the cell and the outside of the cell. Okay, so we establish concentration gradients between two adjacent or regions next to each other. Okay, so this word adjacent means next to each other. Okay, so this is very important for passive transport. What I mean by that, transport that happens due to concentration gradient and doesn't require energy. Okay, so concentration gradients are very important because they are the driving force for the transport of molecules from one concentration to another, and specifically from high concentrations to low concentrations, okay? So this is called diffusion, all right? When a molecule is transported from high concentration to low concentration, it's called diffusion. And there's different mechanisms of diffusion. Some molecules can pass straight through the bilipid layer, phospholipid layer, okay? Because they are non-polar, they are lipid soluble, all right?
Yeah.
So oxygen and carbon dioxide are two particular molecules that can pass straight through the phospholipid bilayer. They don't need any protein molecules, okay, to pass through, because they're lipid soluble. Oxygen and carbon dioxide, and other molecules that are lipid soluble, okay?
Yeah.
So these molecules, they don't require any sort of, I'll just show you here, okay? So here it just passes straight through the plasma membrane, okay?
Yes.
This is happening by simple diffusion, okay? So diffusion straight through the lipid layer. These molecules such as oxygen and carbon dioxide, they're non-polar, that means they're not charged, and they're soluble in lipids, so once they reach the lipid layer, they can pass through it, okay?
Yeah.
And an example of that in our body is, when we breathe in air, the air contains oxygen, correct?
Yes.
In our body, the heart pumps the blood. When the blood reaches the tissues take up the oxygen, and by the time it comes back to the heart, it's deoxygenated. Most of the oxygen is gone from it. So in the blood, we have low oxygen. In the air we breathe in, we have lots of oxygen. So remember, we said that molecules move from high concentration to low. So the concentration of oxygen in the air in our lungs is high, while in the blood it's low. So what happens is that at this point, this process happens, okay? Oxygen moves out of the air into the blood, okay, by simple diffusion through the plasma membrane, okay?
Yeah.
Now, the opposite happens for carbon dioxide. The blood coming back to the lungs, okay, is high in carbon dioxide compared to the air, okay? So carbon dioxide will move out of the cells into the lungs, okay? So it's happening by concentration gradient, all right? And it's happening by simple diffusion. There's no need for any transport molecules, okay, like proteins.
Okay, the opposite process in the same way.
Yeah, so carbon dioxide moves the other way in our lungs, okay? So, depending on the location, it can move differently. In the tissues, carbon dioxide moves out of the cells into the blood as well, okay?
And then it
but
goes to the
from the blood into the air,
Yeah.
okay? And oxygen, the opposite happens in the tissues. It moves from the blood into the cells, depending on the location, okay? We will learn more about this in next semester when we're talking about the cardiovascular system in more detail, okay? All right, so molecules can pass through by simple diffusion or directly through the lipid bilayer, or they can have protein channels, okay?
Yes.
Now, here, can you see this? This is a channel. It's just like an opening, like a pipe, okay?
Always open.
A passageway from one side to the other side, like this corridor, okay?
Yeah.
Without this corridor, can you move from that side of the building to there?
No.
You need that channel, okay? So this acts as a channel from the outside of the cell to the inside. And this is important for molecules, small molecules, okay, like water,
Ions.
which are which are polar, okay? Ions which are polar, so they can't pass directly through the bilayer, okay? These molecules that can't pass through the bilayer, that are charged, need a channel protein, okay? Then the other type is facilitated diffusion, okay? So in this case, you got a carrier, okay? A carrier protein. Once molecules, so for example, there's a molecule there, I want to transport it. So usually this is for larger molecules. That's why they can't use these channels, and they're more specific, okay? These are more specific. So what happens is once the molecule binds to it, the channel, it changes its orientation and shape, and then transport the molecule into the cell, and then once it's released it turns back to its original orientation and does the same thing again, okay? Until you get equal number of molecules on each side, what we term as equilibrium, okay? And I'll talk about that in a minute, okay? So that's facilitated transport. We'll get to it and talk about it in more detail when we get to it. So this is just a summary of it, okay? Osmosis is also the diffusion of water, okay? So water moving from high concentration to low concentration, we call that osmosis. Yes.
It's exclusive to water.
Yes. Osmosis is exclusive to water. When water moves from high concentration to low, it's called osmosis. Then we have other types of transport mechanisms which require energy, okay? Active transport, for example. So these require energy. Bulk transport. So bulk means in large quantities, okay? So sometimes, you have these channels and carrier proteins, they can only transport small amount of molecules. But in bulk transport, the cell can bring in, a bacteria. Remember that endocytic vesicle from part 1?
Yes.
Endocytic vesicle, remember it engulfs the bacteria,
Yes.
brings it inside. This is called endocytosis, this process, okay? In this endocytosis. Remember the secretory vesicles that come from the Golgi and secrete molecules out? This is called exocytosis, okay?
Can you repeat that?
So endocytosis is when the cell brings large quantities of molecules or substances into the cell.
Yeah.
Like bacteria, okay? So remember from part 1, the cell engulfs the bacteria and makes a vesicle and it pinches in and brings it into the cell. This process is called endocytosis, okay?
Yeah.
When it takes it out, like the secretory vesicles coming off the Golgi, remember it secretes some molecules out?
Yeah.
The secretory vesicle comes to the membrane, fuses with it, and releases the molecules into the outside.
Yeah.
You remember that?
Yes.
That's called exocytosis. So endo, remember endo, inside. Exo, outside. Okay?
It's all called
These are called bulk transport, these two processes, because bulk means large quantities, okay? They can bring large quantities at one point, at a time.
And active transport uses
Also, this is use energy. Both of these use energy, okay? This is just a summary. We're gonna go into more detail, okay? So just before we move on, so passive transport, it doesn't require energy. It's from high concentration to low concentration, okay? So molecules, here, remember here there's lots of molecules here. Here, there's not many. So net movement of molecules should be in which direction?
To the
downward.
To the inside of the cell, okay?
Yeah.
Because there's low concentration here, high. So diffusion is the movement of molecules from high concentration to low concentration
Unless there's equilibrium.
until it reaches equilibrium. Okay? So this is happening by diffusion. So movement is from molecules of from high concentration until it reaches equilibrium to low concentration. All right?
Yes.
So what equilibrium is shown very good in this example here. So there's a drop of this blue dye or chemical. So after 10 minutes, you see a little bit of diffusion, okay?
Yes.
The dye is moving away from the high concentrated area in both directions. So if you give it enough time, 1 hour, it's diffused even more. After 24 hours, it's diffused more. So if you give it long enough, this whole liquid will turn blue and it'll be like one color blue, okay? Because of equilibrium. Molecules, if you do chemistry, molecules don't like to be, if you put them in a solution, they don't stay in one place. They like to diffuse until there's, equilibrium in that solution. You know, if you put salt in the water, does it stay in one place or it just
No,
dissolves and moves around?
It moves around.
Yeah. So until it diffuses around, it doesn't like to be concentrated in one place in the beaker.
Sometimes it's go to the bottom of the glass.
Once it's slowly over time, if you give it enough time, it will dissolve, okay, and it will move around to the you know.
Yeah.
Does it move because it wants to be an equilibrium
Yeah.
situation?
Yeah, it doesn't. So if there's room for movement, it will. Okay.
Yeah.
Okay. And that's more, you know, physics. You know, when we're talking about the motion of molecules. I don't know. The molecules don't tend to cluster together.
Yeah.
They like to be in a state of what they call low entropy, low energy. I don't know. I mean, you'll learn that in physics, okay?
Okay.
So equilibrium is when the solutions in both regions become equal concentration, and in that state, there is no net movement of molecules. So if a molecule moves into the cell, one has to move out, okay?
Yeah.
So are we good with equilibrium?
Yeah.
All right, so we'll just go through the three different types of passive transport. As I mentioned, so these molecules that diffuse straight through the plasma membrane, through the bilayer, the lipids, okay, the phospholipids, they're non-polar. That means they have no charge. They're neutral, okay?
Yes.
So molecules like oxygen and CO2, okay? So oxygen diffuses into the cells, CO2 moves out of the cell, okay?
Is it non-polar?
CO2 is non-polar, it is neutral, it has no charge, these molecules. So they can pass straight through the lipid because they're lipid soluble, okay? Some other ways, like urea, you know, the cell makes, when it metabolizes, it makes ammonia, okay? And ammonia is toxic to us.
Yeah.
So the liver takes this and converts it to this less harmful thing called urea. But we still need to get rid of this. So this urea is also neutral, non-polar. It can pass through. It can pass through. Yeah, depending on where it is. You know, in the kidneys, it will. You know, out of the blood into the during urine formation, it can come back. You know, to establish concentration gradients. It's important in the urinary system. This urea, okay? We don't need to talk about it now, but the important thing is it's non-polar, it's neutral, it can pass through the plasma
It's
membrane.
lipid soluble.
It's lipid soluble. So molecules that are charged, they have electrical charge, they cannot pass directly through this because they're not soluble in the lipids. So they need transport molecules, okay, like proteins. So diffusion, the second type is diffusion through protein channels. Now, we have two types of channels. One is an open channel that's open all the time, and one that has a gate, okay? So the gated channels are important because the cell can control the movement of molecules. So depending on the condition of the cell, it can open or close this gate. It's important in the nerve cells, the neurons, these gated cells, because it can control the influx of potassium, calcium, and sodium. These are very important molecules in the electrical impulses. And these gated channels, they're important in those type of cells, okay? So the cell can control molecules. It's just like this door, okay? I don't want anybody come into this room. I can just close it. Well, if you want somebody to come, the students, you open it back up, okay?
Yeah.
So it's a gated. Now, open channels are water can pass freely, so it can get in and out of that channel. And these gated channels, like examples are like sodium, potassium, and calcium, okay? So in this case, also the movement is from high concentration to low concentration because it's still diffusion, it's passive, okay?
Okay, this mean ions cannot pass through the open channel?
Well, there is some channels that can allow. They're open channels. Some ions can pass through. but generally, you know, the
Close.
the gated ones, okay?
They take it when they need it.
Yeah. So it doesn't require energy, okay? And the driving force for this is concentration gradients, okay? So high concentration to low until equilibrium is reached.
Not require energy. Huh?
No.
It's
This-
passive.
This is passive. Okay, so the third type, the facilitated diffusion or facilitated transport requires a carrier protein, okay? So it's different to the channel. In this case, the molecules bind to it, this carrier. Once the molecule is bound to it, it changes its orientation or its shape, okay? So it might be facing this way, picks up once the molecule binds to it, changes orientation, shape, and releases the molecule there, then comes back, picks up another one, does the same thing again. So it's helping the molecule to move across the plasma membrane. So facilitate it means helping something, okay? Facilitating something, you're helping, okay? You're helping for something to happen, okay? So that's what the word facilitated means, okay? So facilitated transport is when a carrier helps one molecule to pass through the plasma membrane through concentration gradients.
Only to pass or also to
So move. So it's moving, okay? It's transporting it from into the cell or out of the cell, okay? So once enough molecules are bound to this, it changes its conformation or its orientation, releases the molecules, and then returns back. Does the same thing again, and this is happening by concentration gradients, okay? So it's using the driving force of the gradient until equilibrium is established. And these are more specific, okay? So they can be specific for glucose or other simple sugars or amino acids, okay? So these are highly selective or specific to particular molecules, all right? And it doesn't require energy, okay? Because it's using the concentration gradient. So the three types, straight through the bilayer. These are molecules that are non-polar, like oxygen, carbon dioxide, or they can pass through channels, okay? Gated or non-gated, like water and sodium, potassium, and calcium. Because they're charged molecules, they can't pass through the plasma, the bilayer directly, or they can be facilitated, where they bind a carrier, and the carrier changes its orientation to transport molecules across the plasma membrane, and this is more selective, molecules like glucose, simple sugar, amino acids. All of these three are happening by diffusion, passive diffusion, no energy is required, and it's happening because there is a concentration gradient. Got that?
Got that.
Yes.
Great. No questions on that?
No.
All right, so let's move on to the next lesson.
That was
Osmosis,
osmosis.
that's what we're talking about now.
Okay. It's a different type, not diffusion or.
It's diffusion, but it's in the movement of water, okay? It's specifically for water. It's happening through these channels, okay? But the process of the movement of water is termed osmosis, okay? Yes. This is HM12, correct?
Yes.
Can you mark your names? So osmosis is, remember, the plasma membrane is highly permeable to water, okay? Because of those channels. So water can move in and out easily. Water, because of the channels, okay? So, but other molecules, they're not so permeable to water, okay? So the membrane is selective, but for water, it's highly permeable. I mean, there is some exceptions in some regions in the kidneys. Some regions are less permeable to water, but that is for the purpose of urine formation, okay? But in general, most cells are highly permeable to water, but less permeable to other
Ions.
molecules or ions, okay? And this movement of water, net movement of water is termed osmosis. So water moving from high concentration to low concentration, okay?
The water, not the
No,
ions.
not the
or-
ions, the water we're talking about. So in a hypotonic solution, water is more, okay, than ions. So if you put some cells in hypotonic solution, where will the water move?
Okay.
Hypotonic. Remember hypotonic? We said
Yeah, higher.
less ions,
Less,
Yeah.
more water.
Yeah.
So remember when we put the blood cells into a hypotonic solution,
It's shrink.
It goes into the
No,
blood.
It goes into the cell.
No, it's
it
lysis.
ruptured because the water moves from the outside into the cell. Remember?
Yeah.
Hypo, okay?
And then it explodes.
So it's happening. How is the water moving? It's moving by osmosis into the cell, okay?
Ah, yes.
Okay? So the water moves into the cell by osmosis. So it's happening through a selectively permeable membrane, like the cell membrane, and it's moving from highest water concentration, okay? So in the highest water concentration, the concentration of solute is low. To low concentration of water, where the concentration of solute is high. So it's moving from a hypotonic to a hypertonic solution. All right. So water movement from low solute to high solute regions. Correct? You got that? Any questions? So that's what osmosis is. Movement of water from high concentration of water to low concentration through a selectively permeable membrane, like the plasma membrane. And we did this experiment and we saw it. Yes, it's true. Remember, the cells shrink in hypertonic solution. When you put them in a hypotonic, they burst.
Eruptions, yeah.
Okay, when you put them in isotonic, nothing happens because there's no osmosis happening because it's in
This
equilibrium.
process is called osmosis.
Yes, the movement of water.
So basically, the smoothest movement of water from hypotonic solution to hypertonic solution.
Yeah.
Hypertonic to hypertonic.
Yeah. So this experiment will show you another way how, and we already did that experiment in the lab, but
Yeah.
here's another one. You see, there's a selectively permeable membrane there. Okay. Only so there's glucose here in this solution, and this one is pure water. Okay. So which one is hypertonic?
Hyper is the left
The
one.
lactic glucose.
So this one is hypertonic. Okay. It's got more solute. This one is.
Hyper.
Which way should water move?
To the left.
To the left, correct?
Yeah.
Okay, so water should move from this region where high water concentration to low water. So this happens through that selectively permeable membrane. This water movement happens until you get this osmotic pressure in this tube forcing, equalizing with the movement of water, okay? So the osmotic pressure equals osmosis. So you see this water level?
Yes.
It's having a force downwards, okay? This force equals the osmosis. So this is called the osmotic pressure. When those two equal, the net movement of water, it'll reach
Stops.
equilibrium, okay? So we got that?
Yes.
So this is osmotic pressure. You know, you have this tube here sitting,
Yeah.
and this water is pushing down. Okay, there's a net force of the water. What we term osmotic pressure.
Ah.
So water will continue to move by osmosis until the osmotic pressure is opposing it. So this here, this level above that water level is termed the osmotic pressure. It's having a force against osmosis, okay? And when that two equals, there's no net movement of water. If
Equilibrium.
water moves this way, water molecule will move back into here. We reach equilibrium because osmotic pressure is opposing osmosis.
This brings equilibrium.
Yeah, it's reaching equilibrium. So osmotic pressure is the fluid pressure, okay? You know, you'll learn this in physics or chemistry, the fluid pressure. It's opposing, okay? There's a force opposing osmosis, okay? So the osmotic pressure is the force exactly opposing osmosis. And that's when there will be no net movement of water, it will be equilibrium. Is that? Anybody didn't understand that? So remember, water is moving this way, correct?
Yeah.
It'll continue to move this way until this osmotic pressure equals exactly the osmosis. And then this pressure will oppose osmosis, okay? And there will be no net movement of water.
It's reached equilibrium.
It will reach equilibrium. So that's what's happening.
And it's called osmotic.
Osmotic pressure. This is what's opposing it, alright? So in that case, we saw that there was a semi-permeable membrane separating the two solutions. Water diffuses, we said, from the right to the left because this one is hypotonic and this is hypertonic. From high concentration of water to low, and this, you see this chamber here, the water rises as osmosis is happening. It'll rise until osmotic pressure opposes osmosis, okay? So in this state, it'll reach equilibrium. When it reaches equilibrium, there will be no net movement of water to each side, okay? Got that?
Yeah.
So the osmotic pressure opposing osmosis. Alright, so this one you've already studied in the lab, so it should be easy to understand. We talk about the tonicity. The tonicity is the concentration of solutes in two fluids, okay? So in this case, in physiological scenario in the cell, we have the membrane separating the outside from the inside of the cell. So we have two regions. We can establish tonicity there, okay? So tonicity is the relative concentration between two different regions, okay? Adjacent regions. Yeah. So when we look at a hypertonic, we said it has more solute, less water. Isotonic equal hypotonic, more water, less solute, okay? And pure water, which has no solutes, is the most hypotonic solution. You know, if you go to the doctor, he will not inject you with pure water and medicine, you know. He will put it into like saline, 0.9% saline, which contains sodium chloride.
Yes.
Because it's 0.9%, 0.9% sodium chloride is isotonic to our blood, okay? So when he injects that with any medicine antibiotic that he dissolves in it, it's isotonic. It won't affect our blood cells. It'll probably kill you if you put a drip with pure water, because you're changing the tonicity, okay? You'll make the blood more hypotonic if you put pure water.
So they'll pump.
So they will use saline, what we call saline, which is 0.0. Remember in the lab we use saline, 0.9%. This water with 0.9% sodium chloride, salt. Yeah. You used it. Yeah. The isotonic solution. It was in a little bottle. Maybe you missed it. Yeah. So isotonic is the same concentration in and out of the cell, okay, of solute. So remember that water moves from hypotonic to hypertonic, okay? So water will move this way, all right? You have more solutes, less solutes in hypertonic. Water will move that way, okay? So water will move by what process? Osmosis, huh? Osmosis. So water will move from osmosis, by osmosis, from hypotonic to a hypotonic solution. And in isotonic solution, do you have movement of water? No. If molecules move in, the same amount have to move out. It's equilibrium, okay?
There are movement by tonicity?
Yes. This is, yeah. We're talking about here, when we're talking about tonicity, we're talking movement of water, okay? Here?
Tonicity is movement of water
Yes.
or
Movement of water from concentrations. So in this case, water diffused from hypotonic to hypertonic, okay?
It's both of them, osmosis and tonicity, it's movement of water.
Well, so well,
One related to water.
one is
solute.
tonicity is talking about the concentrations, okay? Osmosis is talking about the movement, okay? So tonicity, hypertonic, more
More solute.
more solid. Hypotonic,
More
more water. So this is just talking about the concentration of solutes, okay? While osmosis is talking about the movement of water. Is that clear?
Yes.
How are we going for time?
We still have
10
10
minutes.
I think we can finish. Finish the list.
Okay. Yeah, we should be able to finish that. So, yes,
I think Evan.
everybody's here? Everybody's here? One missing. Who's that? Ahmad Muhammad. He's not here?
He got out of the college.
He's got out of the college?
Yeah.
Okay. Alright. Everybody's here. So we said iso means equal, okay? So there's equal concentration between the intracellular fluid, okay? So the cell has intracellular means the inside of the cell, okay? And extracellular means outside. So extracellular is outside of the cell, intracellular inside. So when the concentration of the solutes are the same, it's isotonic, the solutions. Both solutions are isotonic. And this is how our cells maintain the volume, okay? So they don't, burst or they don't shrink. That's why in our body we need to maintain some isotonic conditions, all right? So water cannot, leave the cell and the cells shrink, or the get inside the cells and burst. So we need to maintain that. So this is regulatory, so it's regulated, it's controlled, okay? It's a control mechanism by the body to maintain this isotonic conditions always. So we don't get, the cells dehydrating and shrinking, or they're too much hydrated and they burst. So the cell is always controlling this isotonic conditions in your body. All right. And this is the experiment we did in the lab. Remember, we had the three different beakers or test tubes. We put three different solutions and we needed to identify which solution was what. So we put the cells in them, and in this here, what's happening to the cell here? Nothing's happening to it. It's maintaining its normal cell. So you see these arrows, it's saying that as water moves in, the same amount of moves out, okay? So what sort of solution would that be? Isotonic, okay? In the second solution, we added the cells. We found that the cells are shrinking. So water is moving, more water is moving out of the cell than coming in. So the cells shrink. So what does that mean? What's the concentration of solute here?
Hypertonic.
Hypertonic, okay? So hypertonic, so inside the cell would be?
More.
More water in it, it will be hypotonic inside, correct?
Yes.
Inside the cell will be hypertonic, so water will move from the cell to the outside, the cell shrinks. So we know that that solution is hypertonic, okay? And then we put the cells in a third solution. We found that they are growing and nearly about to burst, okay? So they become enlarged. So we find that water is moving into the,
More than
so what's the solution here?
hypertonic.
Hypotonic. More, it's like pure water. So water likes to move from high concentration to low concentration water, or low concentration solute to high concentration of solute. So you get movement of water into the cell enlarges and bursts, okay, because of that. And you've done this experiment in the lab.
It's a red blood cell.
Yeah, so red blood cells. That's what RBC is. So this is the effect of tonicity on the blood cells, and we did this experiment on the lab.
So for the hypotonic, the small arrow refers that a small amount of water is getting out. Or?
It's just saying that the amount of water leaving the cell is far less than the amount coming in, okay?
But there is a percentage.
There might
that
be
goes.
some water moving out, but the net movement of water is into the cell. That's why this big arrow is showing, and the cell enlarges and bursts. The opposite is here. The net movement is out of the cell compared to the amount coming in, okay? That's why the cell shrinks. Well, that's repeating the same thing I just said. Any questions on that? Everything is clear?
So,
Are we behind on any lessons here? We didn't take that too quickly, or was it good?
No, that was good.
That's good.
yeah.
That's good. All right. I've already mentioned this slide. So there's no questions. We'll, inshallah, tomorrow we'll try to do a Kahoot on these things. Okay?
On this subject, the lesson